Hello everyone, nice to talk to you all. Today we're going to continue our topics on the fundamentals of environmental engineering and science, and specifically we're going to learn about the activated sludge. So let's do a quick recap of our last class. So in last class we went through quite many wastewater treatment processes. For example, for the pre-treatment we talked about the bar racks, the grid chambers, comminutors, and equalization tanks. And furthermore, for the primary treatment, we mentioned that we typically use a primary clarifier, which is using the mechanism of sedimentation. And because of that, we need to use the surface overflow rate as the principal design parameter for the primary treatment. And further, for the secondary treatment, we didn't go into details on how this treatment is done, but we mentioned that we're going to use the microorganisms to oxidize the extra BOD in the water. And we mentioned something about the metabolism, and this is going to be used in activated sludge. And we mentioned different types of the decomposition mechanisms, including aerobic, anoxic, and anaerobic. So we also learned about the growth kinetics and specifically mentioning the Monod equation. So let's do a quick question here. So for an activated sludge process, people observe that the bacterial growth rate is normally lower than the calculated values from the Monod equation, and it's asking why. So we need to mention that for the Monod equation, we don't consider the decay of the bacteria. And because of that, we need to choose B here, because Monod equation just consider the growth rate as a function of the food supply. So it doesn't consider any death of the bacteria or microorganisms. Okay, so let's look at the new contents of this class. So in this class, we're mainly going to focus on the activated sludge process, which is a very important process that can be used in the design of the wastewater treatment plant and also the secondary water treatment processes. So this process is basically happening after the primary settling tank. So we mentioned that this is a primary treatment of the contaminants in the wastewater. So what happens after is that the effluent is going to go through a aeration tank where we have a lot of pipes at the bottom that's bubbling the air through. So by bubbling this air, then the existing bacteria are going to use this oxygen and then oxidize those BOD. Okay, and then as we know, since there are relatively high value of the BOD, then these bacteria or microorganisms can grow by themselves and their population can increase. So by increasing the population, we're basically converting all of these BOD into solids. And after we get these solids, we will introduce the effluent into a secondary settling tank, where it's using the gravity of the suspended solids to further reduce or to further clarify the water. So similar to the primary settling tank, those suspended solids are going to settle down to the bottom of this settling tank. And then we know that since a lot of these microorganisms are already populated themselves, so the settling particles or the settled particles is going to mainly be composed of the microorganisms. And that's why we call these solids as the activated sludge. So although it's a sludge, but it contains a lot of microorganisms. And these activated sludge can be used back to introduce into the aeration tank. So to basically enhance the concentration of the microorganisms. So as we have mentioned, what happened here, or the bacteria existing in the wastewater, are the bacteria just existing in the nature, right? So by bumping up the concentration of these microorganisms, we're simulating the natural processes, but we're trying to process the water with a faster speed. And because of that, we need to bubble a lot of air inside, because the oxidation of the BOD or oxidation of these organics are going to need or require a lot of oxygen. So after the secondary settling tank, then these effluent, or which is a major component of the sewage, are going to go inside the disinfection, because we want to kill all of these bacteria that's growing through the aeration tank. And then further we can emit into the environment there. Okay, so what happens in the aeration tank, or is basically a reactor, is that there is a coexistence of the bacteria and pollutants, and then inside the aeration tank there is mixing and aeration, which means that there's going to be a better contact between the microorganisms and also the BOD sources, or the organics, and then through these aeration processes or aerobic decomposition process, we're going to degrade the organics in the water. So after the aeration tank, the effluent will be introduced into this secondary tank here. So that can achieve the solid-liquid separation, which means that we can extract all of these bacteria or microorganisms out of the processed water. So here we are going to define quite a lot of parameters. So here we will know a new definition, which is called the mixed liquor. So the mixed liquor is just a mixture that's in the aeration tank, because if you imagine, the aeration tank is going to compose of mainly liquids, right? So inside this liquid there are suspended particles and also bacteria, and it's a mixture of what's emitted from the primary cycling tank, and also the return sludge that's collected from the secondary cycling tank. Okay, so after we mix them together, they exist in the liquid phase, and that's why we call them mixed liquor. So because of that, the suspended particles or suspended solids in the mixed liquor is called the MLSS. Okay, that's the mixed liquor suspended solids. So we also define a parameter that's called the MLVSS, where V here is the volatile. So we use the volatile here to represent the microorganisms. So when I mention about the MLVSS, which is the mixed liquor volatile suspended solids, they just mean the solids that's existing in the microorganisms, or it's just the microorganism concentration in the mixed liquor. Okay, so because of that, the MLSS is always going to be larger than MLVSS, mainly because there are non-bacterial suspended solids in the effluent coming from the primary settling tank, right? So those are maybe rocks or salts. So because of that, the MLVSS, which is just for the bacteria or microorganisms, are going to be lower than the MLSS. Okay. So here we'll further expand more on the definitions or the design parameters of an aeration tank. So we mentioned that the MLSS is basically the mixed liquor suspended solid concentration, and typically this MLSS will have a concentration between 2,000 to 4,000 milligram per liter. And we should notice that among these suspended solids, more than half of them are actually microorganisms. And so the higher the MLSS, typically the higher the MLSS, then the smaller the volume we will require for the aeration tank, mainly because if we can process more, or if we can process more higher BOD or more polluted species in the water, organics in the water, then we just need a smaller aeration tank there. But this maximum concentration is limited by quite a lot of parameters. So first, we cannot bump up this concentration by infinitely high, mainly because the oxidation of these organics are going to require oxygen. And there is no easy way to bubble in a lot of this air into the aeration tank. Okay, so there's this maximum concentration is going to be limited by the oxygen. So on the other hand, this is also limited by the clarification capacity of this secondary settling tank. Okay, because if we just produce too much suspended solids, or if we just grow too much microorganisms inside this aeration tank, then that's going to require longer time for us to settle down these solids here. And because of that, that will limit the size, or that is limited by the size of the secondary settling tank here. So that's why we cannot bump up this MLSS concentration to a very high value here. So yeah, we have gone through this slide here. And then the second important parameter is the dissolved oxygen concentration in the aeration tank. So we want enough oxygen in this aeration tank, mainly because the oxygen is basically the fuel or the material that's needed by these microorganisms to oxidize the organics there. So the oxygen concentration or the DO concentration have to be sufficient for enough or sufficient BOD removal. And we normally set this concentration to around 2 milligram per liter. And you can see that earlier in our class, when we talk about the river water quality, we mentioned that the saturation dissolved oxygen concentration is around 8, right? Under normal temperatures, 8 to 10, actually. So this concentration, dissolved oxygen concentration, is actually relatively low here. But and also there are some other parameters that's limiting the concentration of the dissolved oxygen. We cannot make it too high, mainly because if the concentration is too high there, then that means we need to bump in a lot of oxygen or air into the aeration tank, and that's going to cause an energy waste. And typically people realize that 2 milligram per liter is already enough to grow these microorganisms, to consume the organics in the water. And further, if the oxygen concentration is too high, then we're going to prefer, or the growth of these filament bacteria is going to be preferred. So what that happens is that, what that means is that there are going to be different types of bacteria that can grow in the aeration tank. So what people discover is that if the oxygen concentration is too high, then these filament bacteria, which means that they are going to grow in the branch, in the structure of the branches, it's going to, or these types of bacteria are going to be preferred at the high dissolved oxygen concentration. So what that means is that once we form these suspended solids or these bacteria through the aeration tank, when we're trying to settle down these bacteria, then that's going to be more difficult for these branched bacteria to settle down, mainly because the drag force on them are going to be higher. And as a comparison, if we can grow some bacteria that directly has the spherical shape, then that's going to be easier for them to settle down to the base of the settling tank. So that's the reason why we can't bump up the dissolved oxygen concentration too high. But we also cannot set this DO concentration too low, because once that's too low, then there won't be sufficient oxygen to oxidize these organics in the aeration tank, which means that we cannot remove these BOD sufficiently. And also, if the concentration of the oxygen is too low, then as we learned in our last class, the anaerobic oxidation or the anaerobic decomposition is going to be preferred. And we mentioned that the anaerobic decomposition are going to generate a lot of volatile organic species. That's going to generate a lot of odors. And if we don't want complaints of the neighbors nearby the wastewater treatment plant, we have to maintain the DO concentration at a relatively high value. So the other important parameter in terms of design for the activated sludge or this secondary wastewater treatment process is the hydraulic retention time or the residence time of the water in the aeration tank. Okay, so if we draw out that geometry again, so this is aeration tank, right? Then we have the secondary settling tank. Okay, secondary settling tank. And then we mentioned that some fraction of the solids are going to go back. We call this as the return sludge. And then, and actually the return sludge is not the major flow of the wastewater. So most of them are still going to be emitted from the secondary settling tank here. And also at the same time, there is going to be some return sludge that are wasted. Okay, so we can call this as wasted sludge. This is return sludge. And this is a main flow, main emitting flow, or main effluent. Okay, so the hydraulic retention time is just the time of the water in this activated tank here, or aeration tank here. And typically this hydraulic retention time is around 4 to 24 hours. That's really depending on the total flow rate or the processed amount of the wastewater for a specific wastewater treatment plant. And it is also quite easy to calculate this retention time, which is just the volume of the aeration tank divided by the total flow rate of the effluent. So we also want to guarantee that there is enough retention time. And typically people want to mention this is because for a lot of wastewater treatment plant, the storm water and the sewage water, they're mixed. So what that means is that during the storm time, then when we're going to generate a lot of storm water, then that's going to bump up the flow rate into the aeration tank significantly. So if we have a very large value of this total flow rate, then the hydraulic retention time will decrease. So if that decreases, then that simply means that there won't be enough time for the bacteria to oxidize the organics that's in the water. Okay, so that's going to reduce the quality of the emitted water coming from the wastewater treatment plant. So we really want to guarantee that there is enough retention time for the operation of the active sludge process. So there's another time involved, which is called the solid retention time. So the solid retention time is just the time that a bacteria spends during this cycling process. So as you can see, for the hydraulic retention time, there's just one time going through, right? So the water just need to go through one time. But for these bacteria, they're just cycling here again and again. So because of that, the solid retention time, or the SRT, is typically 2 to 8 days. So you can compare that against the hydraulic retention time, which is 4 to 24 hours, right? So we need this long time, and also this is needed for the BOD removal. And to calculate this solid retention time, so we typically use the theta C to represent this value here. So basically, we can use this equation to calculate that, although it might look quite complex here. So what happens is that we are just going to use the mass of all the solids here. So basically, all mass of all the solids that's in this aeration tank divided by the flow rate of these solids through the main effluent and also through the wasted stream here. Okay, so because you can see that the only outlet of all of these bacteria is just these two routes. As in, let's say, if we just define a system of this whole activated sludge process, then you can see that the only outlet for these bacteria is just through the main effluent and the waste stream, right? So because of that, we can just use the concentration of the suspended solids that's in this mixed liquor multiplied by the volume of the aeration tank, which is the mass of all of these suspended solids, and then divide that by the flow rate of the suspended solids that's going through the effluent, main effluent, and also going through this wasted stream here. Okay, so by using this equation, we can calculate what is the solid retention time. So there's also requirements for the solid retention time. It can't be too low, because if that's too low, then what that means is that the bacteria are not going to spend enough time in this system. So if they're, what that means is that if they're not going to spend enough time, they're just going to get dumped out of the system, right? So that will reduce the growth time for the bacteria, which means that we cannot grow enough of the return sludge, or enough of the bacteria to further oxidize the organics that's in the wastewater. And also we don't want this to be too high, because if this is too high, then we're going to generate a lot of these bacteria again. So it's similar to the mechanism of the MLSS here, because if this mixed liquor suspended solid concentration is too high, then that's going to affect the clarification capacity, right? So we mentioned that this is limited by the size of the settling basin. What that means is we're generating too much bacteria, and it's too difficult for us to remove all of them. So similarly here, we cannot set the SRT to be too long, because if the retention time of the solid is too long, then we're generating too much bacteria, and there's going to be a poor settling of all of these microorganisms. Okay? So another important parameter for the design of the activated sludge is the F over M, which is the food to mass ratio. So so here you can think of the organics that's in the wastewater as the food, and the mass here just means the mass of the bacteria. Okay, so this means this is a parameter that controls the ratio between the organics that's introduced into this aeration tank and the bacteria that's growing within this activation tank. Okay, so you can think of this whole oxidation process as the bacteria or microorganisms getting food from the waste stream of the water. Okay, so so basically that is a ratio of the influence of the BOD per day to the mass of the microorganisms. So it's typically in the normal range of 0.2 to 0.5. So you can do an analogy to a person here. Okay, so this is basically the food that we eat every day over the mass or over the weight of us, right? And you can see that for these bacteria, they can grow their weight by 20 to 50% per day, which is amazing, right? So as a person, or as I guess most of the animals will not be able to have this capacity to grow their weight there. Okay, so we cannot set this F over M value to be too high, because on one hand, these microorganisms cannot handle this amount of food. So although they like the food, but they cannot handle all of them, simply because they cannot grow infinitely. And what that means is that there will be remaining BOD that's going out of the aeration tank and then further emitted through the effluent. So we don't want this F over M to be too high. And also, the reason why we don't want it to be too high is that's going to cause a process called the viscous bulking. So you can think of this as a bacteria that grows too fat. Okay, so they eat too much food, and then basically their volume will become so large, which we call them as the bulking, right? And then after these bacteria grow so large, then their density basically remains the same. Okay, so that's going to cause the difficulties in the settling of these bacteria. Okay, simply because their size become too large and it's difficult for them to settle down here. And also we don't want this value to be too low, because if the F over M is too low, and then it's going to cause this increase or promote the growth of the filament bacteria again, and this is again causing the poor settling. So you can see that there are a lot of parameters that we need to pay attention to when we're trying to design these activated sludge process. Okay, so we'll also go through some exercise problems to get you more familiarized with these parameters. Okay, so let's go through these definitions again. So for an aeration tank, which is used for the secondary treatment process, there's a term called the volumetric loading, which is basically how much BOD we're introducing into the aeration tank. And this here is also dividing by the unit volume of the aeration tank. Okay, so you can think of this as the food that's being introduced into the tank here. So there's a term called the food to microorganisms ratio. Okay, so this is basically the F over M ratio. Okay, so there's also a term called the aeration period. So to be just notice the aeration period is just the hydraulic retention time, or the HRT. So this really depends on which definition, exact definition we're using. The aeration time is basically the hydraulic retention time. And for the mixed liquors suspended solids, they are typically in the range of 1,500 to 3,000 milligram per liter, and the dissolved oxygen concentration is around 2 milligram per liter. So here I'm going to show the process for the activated sludge process again. So basically here, we're going to introduce the effluent that's coming from the primary settler, primary clarifier. We're going to introduce the effluent that's coming from the primary clarifier into the aeration tank. So in the aeration tank, this is a completely mixed reactor here, okay? So the bacteria are going to grow because we provide the food there, and then the BOD value are going to drop, okay? So because of that, we're going to basically form more mass or form more suspended solids because we're converting those dissolved organics into the bacteria, which are solid themselves. And then we're going to introduce them into this secondary settling tank, where the heavier particles or the heavier bacteria are going to settle down to the base of this settling tank. So the majority of the water are going to go through this exhaust or the effluent through the settling tank. And we mentioned that for these settled solids, they're going to be mainly composed of the microorganisms, and we can use some portion of these microorganisms to introduce them back into the aeration tank so that we can further enhance the reaction rate or the oxidation rate of these microorganisms. Okay, so basically for these sludge here, we're splitting them out into two parts. Okay, so one part is the return sludge that's going back to the aeration tank. The other part is the waste sludge, which means that we cannot, basically we cannot return all of these sludge back. Otherwise, the concentration of these bacteria here is too high. So a portion of them are just going to be dumped directly as a sludge or waste sludge that's coming out of the system. Okay, so as we can see, if we choose this system that's in the dashed box here, then there are several mass balances we can build up. So the first mass balance, is just the mass balance for water, okay, or the volume balance of water, because the water volume is not going to change, right? So if the inflow rate of the wastewater is Q here, then we know that the flow rate of the effluent and the flow rate of the waste are going to sum up to be exactly equal to the inflow rate, right? So here you can see that the waste stream has a flow rate of QW, and then the effluent stream are going to have a flow rate of Q minus QW. So basically, the first mass balance is just for the water, and the second mass balance is going to be the suspended solids. So what happens here is that if we consider what is the suspended solids within this aeration tank, so if we know that this MLSS, which is the concentration of the suspended solid in the mixed liquor, right? So if we multiply this concentration by its volume, then we can calculate what is the total mass of these suspended solids in here. So if you trace what is the ultimate directions of these suspended solids, so you can see that a proportion of them are going to be emitted through the effluent, a proportion of them are going to settle down in the tank, right? And then some of them are going back into the aeration tank, right? Some of them are going to get emitted as a waste. So if we label them out, this is 1, this is route 2, this is route 3, then the suspended solids are going to be conserved through this three routes here. So 1 plus 2 plus 3. Okay. So further, we can use the concentrations or the flow rates of these suspended solids here, and to build up this mass conservation or the mass balance. So we can go through some practice later on here. So we mentioned that for the design of the activated sludge process, there's an important parameter called the sludge retention time. So there are also other names for this term. for example, the sludge age or mean cell residence time, the MCRT. Okay, so to conserve or to keep all of these terms unified. So here, starting from now, I'm just going to use the sludge retention time to refer to this specific time here. So again, the sludge retention time is the average time of the microorganisms that live in the aeration tank. It is different from the hydraulic retention time or the aeration period here. Okay, so it is also the time for the microorganisms to degrade the pollutants here. And again, I'm showing the calculation equation here. So we can use the mass of the suspended solids that's in the aeration tank and divide by the mass of the suspended solids or the flow rate of the suspended solid through route 1 and route 3 because they're leaving this whole system here. Okay. So the sludge retention time, or the SRT, is critical to the effluent quality, mainly because if we have a low sludge retention time, that means a high food over mass ratio or a high soluble BOD in the effluent. Okay, so if we have a low sludge retention time, or the SRT, it also indicates that the microorganisms are young and active, and they're difficult to settle down. Okay, and this low SRT may also keep some of the slow-growing bacteria, such as nitrification bacteria, in the system. Okay, and also we don't want a high sludge retention time, mainly because if there is a high residence time of the bacteria in the system, then that will cause this endogenous respiration, which means that the bacteria are going to decay themselves, which means that the system needs more oxygen and may use more energy to operate. Okay. So, so we talked about the aeration tank, which is majorly used for the oxidation of the organics there. So downstream of the aeration tank, we're going to use a secondary clarifier. So the secondary clarifier mainly is just used for removing these suspended solids that's in the effluent of the aeration tank, right? Because we have grown too much of the microorganisms there, and then we don't want to emit all of these microorganisms directly to the environment. And also we want to use a proportion of these microorganisms to increase the oxidation rate of the organics that's in the wastewater. Okay, so typically for the secondary clarifier, it is operated at an average overflow rate of 600 to 800 gallon per day per square foot. Okay, and the overflow rate at the peak flow are going to be higher. Okay, and the minimum hydraulic retention time is 2 to 3 hours, and the solids loading rate is 40 to 50 pounds per square foot per day. And it is also assuming that 100 to 150% return at the average flow. Okay, so these are probably too much into details regarding the design of the secondary clarifier. So if you're interested in these details, you can go through the textbooks. But here we're just going to use the concepts of these primary clarifiers, right? So again, for the primary clarifiers of the wastewater treatment, the overflow rate, the surface overflow rate, is going to be very important. But for these secondary clarifiers, we also need to consider what is the suspended solid amount, because the mass of these grown microorganisms are going to also affect the performance of these secondary clarifiers. So that's why we want to operate them under a range of the suspended solids there. So because of that, if we look at this question here, so when we design a secondary clarifier, what parameter must be used in addition to those for the primary clarifier design? So we mentioned that for the secondary clarifier, it's mainly targeted at removing the microorganisms that's grown in the aeration tank. So because of that, we need to consider the solids rolling rate in the secondary clarifier. Okay. So here are more detailed information on the sludge production. So basically, in the aeration tank, the bacteria will consume the organic matter and then convert to biomass, which are solids. Okay, so the older the biomass, there will be less the sludge generation. So approximately 1/3 of the influent organic matter, or the BOD, ultimate BOD, is converted into the biomass. And typically the ratio between the MLVSS, again remember that V here represents the bacteria, right? The MLVSS means the mixed liquor volatile suspended solids, which are the mass or the concentration of these microorganisms. And the MLSS is the all of the suspended solids there. So typically they're in the ratio of 0.6 to 0.8. And people can also calculate what is the total MLVSS production that is happening within the aeration tank. And this is just based on the difference of the food concentration or the organics concentration. So the S0 is the inflow organics concentration, and the S here is the outflow concentration of the organics. Okay. So we also need to consider the oxygen demand during the activated sludge process. So we mentioned earlier in the definition of the BOD, which is biochemical oxygen demand, we know that the BOD is always lower than the theoretical oxygen demand, right? This is mainly because a fraction of the organics are actually converted to the bacteria mass itself, right? And because of that, so 1 grams of the bacteria, so typically that represents around 1.4 grams of the oxygen demand, which means that by forming these bacteria, we're actually taking a fraction of the organics that's existing in the water, right? So because of that, we will require less oxygen to oxidize all of the remaining organics there. So this is a very simple way to calculate what is the demand of the oxygen to operate the activated sludge process. So you can see that the mass of the oxygen is calculated by 1.5 multiplied by Q multiplied by S minus S0 minus S. So S0 is the organics concentration that's at the inflow of the aeration tank, while S here is at the outlet of the aeration tank. Okay, so here is S0, here is S, and then Q is the flow rate that's going through the aeration tank. So this has already been so the S here has already been converted into BOD. Okay, so we use this 1.5 here to basically convert the BOD5 into ultimate BOD to consider all of the oxygen that's required. But we know that this concentration of the organics also decreased, not only because of the oxidation, but also because a fraction of them are converted into the bacteria. So that's why we also need to minus 1.42 of the, which represents the conversion of these organics to the bacteria themselves, and then multiply that by the total amount of the bacteria cells in the through this process. Okay. So here I'm going to show a quick example about how to consider all of these parameters, so how to assess all of these parameters during the design of a activated sludge process. So let's say that if we try to design an aeration tank system or the active sludge system, it provides you that the aeration tank volume is 120,000 square foot, cubic foot, right? Which is close to around 0.898 million gallon there. And the wastewater flow is 3.67 million gallon per day. Okay, and the waste sludge flow is 18,900 gallons per day, which is 0.0189 million gallon per day. It provides the mixed liquor suspended solids, and also the suspended solids in the waste sludge. So basically, if we reconstruct that system there, this is the activation tank, right? And then we have the secondary clarifier, and then this is the effluent. And then for the secondary clarifier, we have this return sludge here. See? Return sludge. And then we'll go this way. Okay? So what this already tells us is that we already know the aeration tank volume, right? And also we know the wastewater flow here. Okay, the waste sludge flow is basically the flow here, right? The sludge, a fraction of the sludge is wasted, right? So here is the Q effluent, which is the majority of the flow. Then we also have a flow that's returning back into the aeration tank, which is QR. Okay, so you can see that this problem already provides us with the volume of the aeration tank, the wastewater flow, the waste sludge flow, right? And also the MLSS, which is basically the concentration of the suspended solids that's in this aeration tank, right? And also provides us the suspended solids concentration that's existing in the waste sludge. So basically, if the waste sludge concentration or the suspended solid concentration in the waste sludge is known, then the concentration in here will be the same, right? Because they're the same bunch of the solid sludge here, but they're splitting one stream going to waste, one stream coming back to the return sludge. So it further provides us the influent BOD here, which is at the beginning or downstream of the primary clarifier, and then it provides the effluent BOD and also the effluent SS, or the suspended solid concentration. Okay, so basically, yeah, so this is exactly the same picture that we're talking about here, right? So we have the aeration tank. After the bacteria grow in here, and then they'll settle down, they'll go into a secondary settling tank where the suspended solids are going to settle down, and then further they will split into the return sludge and also the waste sludge, while the majority of the flow is going to go through the effluent. So it wants us to calculate the following parameters, including the return sludge flow and ratio, right? So basically the return sludge flow is just the QR here. So the return sludge ratio is going to be the QR divided by Q. So that's the ratio compared to the total inflow rate. So it also wants us to calculate the BOD loading, which is the total BOD that's basically introduced into the aeration tank here. So we can also calculate we should also calculate the sludge age. So remember, the sludge age is just the total mass of the sludge, total mass of the suspended solids divided by the flow of the suspended solid through the effluent, and the flow of the suspended solids through the waste. Okay. So we should also calculate the aeration period. So aeration period is just the hydraulic retention time. Remember, we can calculate that by just dividing the volume of the aeration tank by the flow through the aeration tank. So we can also calculate the sludge production rate, which is just at the bottom of the secondary clarifier, and also the BOD removal efficiency. Okay, since we already know the BOD concentrations at the effluent and also at the inlet, we can calculate that efficiency too. So let's try to solve these parameters one by one. Okay, so first let's calculate what is the return sludge flow. Okay, so what we need to do is to set up a mass balance. So as we mentioned, there are two mass balances. So first mass balance is the water there. So we know that the wastewater flow is 3.67, right? The waste sludge flow is 0.198. So by setting up this whole activated sludge process as a system, then since we know these two, then we can calculate what is the effluent flow rate there. Okay, so Then we need to set up a mass balance for the suspended solids, which is just looking at this system here. Okay, so we mentioned that the total mass of the suspended solids introduced into the secondary settling tank have to be equal to the suspended solid mass suspended solid mass through these three routes here. So that's why we can set up this equation here. Okay, so since we already know what is the what is the suspended solid concentration in the aeration tank, then we can calculate what is the total suspended solid concentrations here. So that is 3.67, right, which is the flow of the wastewater plus the flow rate of the return sludge, because here we are also introducing some flow rate in here. Okay, so after the mix, they will form a mixed flow that's introduced into this aeration tank, right? So we should use this combined flow rate, multiply that by the MLSS to calculate what is the total mass of these suspended solids. So we introduced that we have to consider these three different routes, right? So there's a branch of the suspended solids going back into the aeration tank, that is the return amount, because we don't know what is the flow rate of the return flow, right? We're trying to solve for this return sludge flow. So we can just use QR and then multiply that by the concentration of the suspended solids that's in this stream here. So we mentioned that the suspended solid in the waste sludge is 11,000 here. And then we also need to consider what is the suspended solid concentration or the mass in the waste, right? We know the flow rate, we know the concentration, we multiply that together. And then we also need to consider the suspended solid mass through this effluent, right? That is the washout. So that is calculated by the flow rate, which is the total flow rate of the wastewater minus the wasted sludge, which is the effluent flow rate, and multiply that by the suspended solid concentration of this stream here, right, which is 26 here. So basically, after we set up this equation, you can see that the QR is the only unknown in this equation. So after rearrange it, we can calculate that the QR is 1 million gallon per day. Okay, so based on that, we can further divide this value by the total inflow or the influent, right, which is 3.767. We can find that the return sludge ratio is around 27%. Okay, so this is how we can calculate the return sludge flow and the ratio. And now we need to look at what is the BOD loading. So the BOD loading is the total amount of BOD that's introduced into the aeration tank. So since we already know the BOD that's in the influent, so since we already know the flow rate of this wastewater, okay, 3.67, and then we can just directly multiply them together. And you notice here that in this problem we're using pounds here. So basically, first we need to convert gallons into liters, and then further convert standard units of the mass into pounds here. So that conversion is done by this 8.34 value here. So basically, by calculating this, we can obtain what is a BOD load or BOD loading, right? And further, we can calculate what is the total MOSS that's existing in the tank. So we can directly use the tank volume, multiply that by the concentration of the suspended solids that's in the activated aeration tank. And again, we're using this conversion of 8.34 to calculate what is the total MOSS in the aeration tank. So further, with these values, we can calculate what is a BOD loading. And then the BOD loading is just this value divided by 120. Okay, so in this way, we can calculate what is the loading per volume of this aeration tank here. And we can also calculate the food over mass ratio based on these calculations, right? BOD is a food, and then the suspended solid mass is the mass of the bacteria. So as we have mentioned, the sludge age is just the total mass of the suspended solids divided by the outflow of the suspended solids, right? So we need to calculate what is the total mass of the microorganism that's in the aeration tank. So since we already know the concentration of 230/50, multiply that by the volume of the aeration tank, and then divide that by the outflow of the suspended solids there. So one of them is the basically the outflow through the effluent, right? 26 multiplied by the effluent flow rate, and the other is just the waste sludge there. So we know that waste sludge has a flow rate of 0.189, and then the concentration is much higher, 11,000 there. So we can calculate what is sludge age. So the aeration period is just the total volume of the aeration tank divided by the flow rate of the wastewater, and then we'll multiply that by 24, mainly because we want to convert the days into hours. Okay, so further, for the sludge production rate, then we can use basically what is the total amount of the sludge, and then to divide that by the total BOD that we introduced into the system. So since we already know what is the BOD in the influent and effluent, we can directly calculate what is the removal efficiency. So you can further look into how we can calculate this when you are trying to solve problems or similar problems here. So here's another question. It's asking what is the sludge age? So based on the sludge age, that is the total amount of the time that the bacteria will spend in the activated sludge process, right? It's different from the hydraulic retention time. So that is, because of that, we need to choose B here, which is the average time of the activated sludge that stays in the system. So due to the time constraint, we'll probably go through these two example problems in our next class. So as you can see, for the design of the activated sludge system, we have to use the mass balance here. So one of the mass balance is for the water, the other is for the suspended solids. So we need to consider different mass balances to further look into these design problems. So maybe we can quickly go through the reviews, a few review questions for this class. So we introduced what are the major unit processes of an activated sludge process. What are the design parameters for the aeration tank? As you can see, there are quite many parameters, and we want to maintain them in the appropriate ranges. So further, what is the sludge age, or the retention time, right? Solid retention time. Why is it important for the effluent quality? And also, what are the major design parameters of a secondary clarifier of an activated sludge process? So we mentioned that we also need to consider the solid loading of the secondary clarifier. Okay, so how do we calculate the sludge production and oxygen demand for the activated sludge process, and also what are the control parameters of the activated sludge process? So that's all for the contents of this class, and feel free to let me know if you have any questions. Thank you.